Hello everybody, today we're going to be talking about viruses. And as we all know by now, the pandemic happened and the coronavirus has changed our lives in a lot of different ways. So that is the same family almost of species that we're going to be talking about, viruses. Viruses are known as the links between the living and the non-living. Why? Because when they enter a living cell, they become activated. And when they're not in a living cell, they just become stationary. They don't function, they don't move, they don't metabolize, they don't do anything. So they're like they're a link between the living and the non-living. It is a very important point here that viruses are not cells. Viruses are not cells, so make sure that you do not ever refer to viruses as cells, because they are not. They are, honestly, we don't know what they are, because we only know that there are links between the living and the non-living, but they are not cells. Next point is that because they are not cells, they are not classified as a prokaryote or a eukaryote. Which makes sense because they're not cells to begin with. Another feature is that they are very, very, very small. So they are really minute. They are way, way smaller than a bacterial cell, and we need electron microscopes to see a virus. Electron microscope. Okay, next point is that they have genetic material. So their genetic material is DNA or RNA, depending on the virus. And they have what is called a capsid. And capsids are protein coats which basically protect the DNA. So protein coats to protect the DNA or RNA. As far as reproduction is concerned, they do not reproduce like bacteria or like any other organism. In fact, they cannot reproduce, let's not say reproduce because it's a little misleading. Let's say they cannot multiply without a host cell. So they, as we said, they're a link between the living and the non-living. So when they are in a living cell, that's the only time they can multiply and grow in number. There are two types of replication processes. One is called a lytic cycle. Oops. In a lytic cycle, the virus binds to the surface of a bacteria or any host cell. I'm just referring to bacteria as an example. But this is the host cell, and this is the virus. And mind you, viruses are way smaller than this size, but just so that we have a idea. So viruses are way smaller compared to the host cell, and they bind to the surface of the host cell, and then they inject their DNA inside of the host cell. They either do this or they just enter the host cell as a whole, as they enter their whole self into the host cell. And then they inject their DNA then into the host cell. This makes the host not realizing that the bacteria is inside its body. The host starts replicating the viral DNA. And after a while, the host cell is filled with these viruses, and that causes cell lysis, which means that the cell basically ruptures and dies, and all the viruses, the replicated viruses, come out of the ruptured cell. So this is cell lysis. It just means that the rupturing of cell walls, of the cell membrane, excuse me. So this is the lytic cycle. This is one way viruses replicate their DNA and multiply themselves, by going into the host cell and then using the host's resources to multiply itself, and then finally, which ends to the host dying and all the multiplied viruses comes out, and then they affect more host cells. The other type of reproduction is called the lysogenic cycle. Over here, just like in the first in the lytic cycle, the virus injects the DNA into the host cell, just like the first one. So the virus injects the DNA into the host cell, and then instead of just having the DNA freely flowing, it binds its DNA, with the DNA of the bacteria, for example. So if this is a plasmid, right, then the let's change the color here. The viral DNA, which is this black DNA, that will attach itself to the plasmid, and then this bacteria, not realizing that it has a viral DNA, it will replicate itself along with that viral strand of DNA. So like everywhere, every single time these cells replicate, there is a copy of the viral DNA inside of every cell. And as the cells increase in number, the viral DNA increase in number. And this is not a problem until now, right? However, if the virus DNA is triggered by an enzyme, or by lack of food or lack of resources. So by, let's say, lack of nutrition or enzymes, then the viral DNA can lyse the whole cell. So then we go back to the lytic cycle from here. If it is triggered by any means, then we go to the lytic cycle, where the cell lysis happens, and then all the DNA go out, and the cell is ruptured. This is okay if it's just one cell, but once there are so many copies of the same bacterial or whatever host cell, if there are so many copies of an RNA, of a viral DNA strand, and if all of them lyse at the same time, there is a mass death of cells. And this can be very deadly for any organism or any community of organisms, or any host cell. So these are like the two types of reproduction or replication cycles that viruses use to replicate themselves. And this is the video. This brings us to the end of the video of talking about viruses. I hope this made some concepts clear, and thank you for watching.